One of the virtual labs we're working on in AP Biology is the enzyme lab, and specifically it looks at the activity of the enzyme catalase in breaking down hydrogen peroxide into oxygen and water. And the setup we're going to use in this virtual pivot lab is the idea that if we collect the oxygen that's produced, we can measure the amount of enzyme activity that has taken place. So the kids are presented essentially with a video that they can control. They can play the video as many times as they want. They can stop it at any time when necessary. But what's nice about this is that they're also able to manipulate parameters like pH and temperature. So unlike a video on YouTube, which is somewhat passive, this allows them to actively go in here and change settings. And another clever feature here is that the pivot setup does not tell them what each of these pHs is. So if they choose pH A, it's a little bit of a mystery as to what that pH might be, and it requires them to figure that out, sort of investigate that on their own. The only clue they get is the color of the liquid in the test tube. So they go to their tools up here, and they bring out their pH chart, and then they can compare that, that color matches most closely with pH 3. Therefore, our pH at this point is pH 3, which is fairly low, and we're able to proceed from there. So the setup of the lab starts by having them, they're being told certain parameters, so they're given a certain amount of instruction to start with for them to figure out the optimal temperature for the enzyme catalase. So they're actually instructed to go to pH C to start with, which is as close to neutral as it could be. Let's load that in there and check it out. So here we have somewhat of a green color that matches up with pH 7, and then they can go ahead and try different temperatures. So the temperature is controlled over here also. They would start with the cold temperature, load that in, and then run the experiment. It's a very brief video that it only takes maybe about 15 seconds, so it's not too time consuming. And then the actual reaction, which you can see here, then is sped up, which can be paused at any time. The instructions tell them to go to 10 seconds, and they need to stop right at 10. So we try to hit it as close to 10 as possible. I'm at 9:50:59 here. Let's see what we can do. So that's about as close as I need to be. I can try and maybe get it a little closer, but if I overshoot a little bit, that's okay also. Let's see here. Okay, let's go back. Try that again. Let's see if we can get right to 10. Ideally, I would be editing these mistakes out, but this is me being a rookie on the virtual lab. The kids probably did a better job with this than I'm doing right now. All right, so I'm at 9:50. I'm going to leave it at that. And what I can see now is how far the water level was pushed down by the oxygen that formed. Here comes the neat part that I really like. It doesn't just tell them how much oxygen formed. A lot of times on virtual labs, it just the computer just tells them, okay, here you got your 5.7 milliliters. Nope, the kids have to actually measure this on their own. So we get a virtual ruler that they then have to line up with the tip of the test tube, and they have to see how many centimeters of oxygen were formed. So we line that up, and then we go down here, 5, 6, 7.6 maybe, and that would then be recorded in the data table. So let's check that out. We minimize this a little bit. What did I say? It was 7.6. I go down to the data table, and I would enter in at temperature 0. We had, I forgot what it was now, 7.6. There we go. 7.6 centimeters. All right, so then we would run the experiment again, and this time change the temperature to the next one, which is a little bit warmer, like room temperature. Load that in, and then run it again. And what's going to end up happening is the kids are going to generate a graph. Now, here we go. Let's just put in some data here of what my estimation will be to save time. But let's say at 20 it produces, I don't know, 10.0. I'm just making things up, at 37 it produces, let's say 5.6, and at, I forgot what the other temperature was here, 75, which is really hot and which hurts the enzyme. So the kids will have to think about what's happening to enzymes at the hot temperatures. The protein gets denatured, and we end up getting 0. All right, so once that's done, they have to go to the graph, and then they have to basically set up their own graph. They have to configure the x-axis by telling the software which of the two variables they want to put in there. Do they want to put temperature in there or oxygen? And then I'll have to know that it's the independent variable, which in this case is temperature. And we configure the y-axis. This would be the dependent variable, which is oxygen. Set that up, and it makes the graph for them. So the one criticism I would have here is that it makes the graph for them. I prefer the kids having to scale their own axes. I prefer the kids having to plot their own point. That's an experience that's valuable for them. Once they get good at that, yes, we can use computers to make the graphs for us, like Google Sheets or something like this. But in this case, it makes the graph, which is okay because I'm going to have them make their own graphs with some of the other data that they're going to collect at a later point. So that's all right to get started because they're still going to have that experience in my class later anyway. All right, well, this is just the beginning of the whole lab. There's more to it. There's also a pH component, and then there's a component that I added, that I customized, which is down here, where they have to measure enzyme rate over time. They have to figure out the optimal temperature first with this initial paradigm. Then they have to figure out the optimal pH, and then use that information and apply that in order to figure out enzyme activity rate over time in part 4. The data from part 4 they would then record and enter into a Google Sheet and then create a graph with this for their lab report.